Going live in 10 seconds.
Hello everyone, and welcome to today's The Scientist Symposium. I'm Nathan Nee, Associate Science Editor for The Scientist, and I will be your MC and moderator for our course of events. Today, our esteemed speakers will explore the limitations and challenges of current SARS CoV-2 qPCR-based testing approaches, their root causes, and what can be learned for the future. Our schedule this morning consists of 3 presentations, followed by a live panel question and answer session. The presentations will be delivered by Dr. Steven Bustin, Jim Huggitt, and Mikhail Kubista. And they will be joined by colleagues Dr. Jo van de Sompere and Dr. Michael Fafel for the Q&A. We encourage you to send us your questions or comments at any point during the symposium, and our panel will address these during the final live Q&A session following the 3 presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our symposium is aided by several sponsors, whom I would like to thank at this time. MilliporeSigma. MilliporeSigma is the life science business of Merck, Darmstadt, Germany, and operates with more than 19,000 employees and 72 manufacturing sites worldwide. Our portfolio spans more than 300,000 products enabling scientific discovery. The company is committed to solving the toughest problems in life science by collaborating with the global scientific community. For more information, visit www.sigmaaldrich.com. PHCBI. PHC Corporation of North America is a subsidiary of PHC Holdings Corporation, Tokyo, Japan, a global leader in development, design, and manufacturing of laboratory equipment for the biopharmaceutical, life sciences, academic, healthcare, and government markets. This includes the space-saving and energy-efficient VIP Eco, TwinGuard, and VIP Series ultra-low temperature freezers, cryogenic and biomedical freezers, pharmacy and high-performance refrigerators, cell culture CO2 and multi-gas incubators, Drosophila plant growth chambers, and cell processing workstations. For more product information, please visit www.phchd.com/us/biomedical. Acro Biosystems. Acro Biosystems is a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics and diagnostic kits. To facilitate research on SARS CoV-2 and help fight the COVID-19 pandemic, Acro Biosystems has developed more than 60 SARS CoV-2 related products, of which 2/3 are recombinant SARS CoV-2 antigens. Acro Biosystems has also developed a series of anti-SARS CoV-2 antibodies, including our unique human-derived neutralizing antibodies. Finally, Acro Biosystems has launched antibody titer assay kits and neutralizing antibody screening kits for vaccine development and drug development. We believe our SARS-CoV-2 related products can make research on SARS-CoV-2 faster and easier. Biosearch Technologies by LGC. Biosearch Technologies is the comprehensive genomics portfolio from LGC, providing products and services for genomic analysis that support mission-critical applications for global customers in molecular diagnostics. We enable our customers by complementing their product development pipeline through our robust research, grade, and cGMP oligonucleotide manufacturing. We also offer expertise in NGS, isothermal, qPCR-based applications, where technologies such as BHQ probes are frequently used in LDTs, ASRs, and in vitro diagnostics. Codex DNA. Codex DNA Inc. is building biology. Creators of the BioXP system, the world's only fully automated gene synthesis platform, and the industry-standard Gibson assembly method, Codex DNA Inc. empowers researchers with the tools they need to rapidly and securely design, code, and create synthetic DNA. Codex DNA Inc. is accelerating advances in the fields of personalized medicine, antibody engineering, vaccine development, drug discovery, and DNA storage. Our sponsors have provided us with some helpful resources related to today's symposium, and we will post these in our handout section located on the right side of your screen throughout our 3 presentations. You can access and download these documents at any time during the webinar. With that, let me introduce our first speaker, Dr. Steven Bustin. Dr. Steven Bustin is Professor of Molecular Medicine at Angela Ruskin University in the United Kingdom. His research interests center around utilizing antibody and PCR-based methods for the development of novel approaches for the early diagnosis of pathogens. Dr. Bustin has authored numerous papers, review articles, and book chapters aimed at improving the reproducibility and robustness of real-time quantitative PCR, including 3 books, A to Z of Quantitative PCR in 2004, The PCR Revolution in 2011, and PCR Technology in 2013. Dr. Bustin was an expert witness advising the UK High Court on qPCR technology in the measles, mumps, rubella vaccine autism class action lawsuit, as well as the MMR trial in Washington, D.C. in 2007. He led an international consortium developing the MIKE minimum information for publication of quantitative real-time PCR experiments guidelines for the use and reporting of qPCR and digital PCR. Finally, Dr. Bustin has extensive editorial involvement as editor-in-chief or as an editorial board member for several peer-reviewed journals. Dr. Bustin, let's make sure your slides are up and running to the audience.
I don't have the icon to share my screen.
Hi, Dr. Bustin. You don't see the screen option at the top?
Ah, there it is. Yes, so I'm sorry.
No problem.
Being stupid again.
No problem. Let's just make sure that that loads to the audience.
Right. Can you see my screen? Do you see my screen?
It's just loading now. Here we go, Dr. Bustin. Go ahead, take it away.
Thank you very much. I'm very grateful to the scientist for giving me the opportunity to present my opinions on COVID-19 testing, but I would like to emphasize that they are my personal views. As a molecular biologist, my interest lies with developing protocols that ensure diagnostic tests are reliable. A positive result should always be due to the presence of a target sequence. The interpretation of the results, especially if they are marginal positives, is a separate issue and requires further examination. Broadly speaking, there are 3 types of test, each one with its distinct advantages and disadvantages. The most commonly used tests target viral RNA, and Rtq-Pcr is the most prevalent. This is due to its flexibility and the ability to detect ongoing infections, and it also offers the promise of extremely rapid and possibly reliable testing. Clearly, it has been the foremost technology that has enabled the testing and tracing paradigm to be implemented. Viral RNA can be detected several days prior to the onset of symptoms and for more than a week thereafter. However, the veracity of testing can be compromised by at least 3 issues. First, during the early and late stages of the disease, viral load is low, with the potential for the test to return a false negative result. Second, false negative results can also rise as a consequence of deficient sampling and RNA extraction procedures, as well as of poor primer design. Of course, together with issues of contamination, careless assay design can also generate false positive results. Third, and this is beyond the scope of my talk today, test results indicating the presence of small amounts of RNA may not be necessarily indicative of the presence of infectious virus. COVID-19 testing has had a very bad press, with most members of the public and, dare I say, politicians and many public health officials ignorant of which test does what each testing methodology's strengths and weaknesses are, and how each test is most effectively employed. And I guess not surprisingly, this has been picked up by the media and is a major contributory factor to the confusion that surrounds the validity of scientific judgment today. And we can see all over the world people protesting and coming up with crazy ideas. Anyway, in Britain, the whole of the testing regimen has been a complete shambles, starting with the initial preparedness, the choice of testing methodologies, the availability of reagents, the testing infrastructure, as well as reporting and follow-up. And this continues to today. When I recently tried to look at our local statistics here in Hertfordshire, I was provided with this information. And as you can see, for most English counties, no data are available. So this kind of tells you what the situation is like 6 months after testing and tracing has been implemented. Botched qPCR testing was first brought to the public's attention when it was used to falsely support an association between the MMR vaccine, measles virus, and autism. And so my experience in exposing the flawed use, interpretation, and reporting of Rtq-Pcr was the catalyst for the development of the MICI guidelines, which we at the time hoped would lead to more appropriate use of this technology. However, when we surveyed qPCR practices a few days later, it was clear that the majority of researchers showed little interest in its proper use. Unsuitable RT-PCR testing was also evident from my appraisal of its use for forensic applications, where poor methodology, protocols, and flawed primer designs combine to make results aiming to identify the origin of human tissue unreliable, at the very least. In both spheres, the major pitfall concerns the generation of false positive results, either because of contamination or because of careless assay design. Unfortunately, it appeared that pre-2020, most researchers, authors of journals, and editors, authors or journal editors, did not seem particularly concerned about ensuring transparency of reporting or the validity of their conclusions. Today, I guess the impact of the well-known but ignored inadequacies of these technologies have become life or death issues, and suddenly people are interested in obtaining more reliable, robust, and accurate testing results. And surprise, surprise, the issue of false positive again has raised its ugly head, this time in combination with the dilemma posed by false negative results. So testing is actually a multi-step process, and together these contribute to the success of the diagnostic test, but they can lead to its failure if mismanaged individually. In addition, of course, to all of this, there's a major element is time, and a rapid return of results is an essential component of the testing tracing paradigm. Because obviously, if you test as the United States and you don't get a result until a week later, then it's pretty pointless testing. So I would like to deal with three of the issues, these issues, and these are issues we've been looking at ourselves over the last 6 months and illustrate our approach to improving or potentially improving the RT-PCR testing procedure. Our solutions involve, firstly, simplifying the sample collection. Secondly, maximizing the specificity of the test, which also leads to the increased sensitivity. And finally, developing a protocol to speed up the time it takes to complete a PCR run. Our workflow extracts RNA from saliva, and it combines the extraction, enrichment, and reverse transcription into a single step to generate DNA. We have developed a multiplex assay that detects 3 viral genes and 1 human gene, plus an artificial extraction and inhibition control, which we subject to fast PCR. And this combines elements of real-time and endpoint detection. Runtime is instrument dependent, but we ultimately aim to use extreme PCR to reduce the PCR time to below 30 seconds. We also have been testing for the D614G mutation, but since April, every single sample we've tested has been a mutant. So in the UK, well, at least in Essex, in Southeast England, all SARS CoV-2 isolates contain the D614G mutation. And we report results with a confidence value rather than as a quantification cycle, because we believe that CQ reporting is wholly inappropriate. So my first topic deals with a process that comprises an essential component of successful diagnostic testing, and it should be as simple, robust, and fast as possible. We've known since February that epithelial cells in the mouth, tongue, and salivary glands express high levels of angiotensin-converting enzyme 2 receptor, which is the main host receptor for SARS-CoV-2. So given all the challenges that surround swabbing, we decided very early on to explore the use of saliva as a simple, fast, and easy to implement alternative. And to our surprise, we found that RT reactions can function even if they contain significant amount of saliva. And just as an aside, in all our testing, we found that the Dpcr prime script was the best RT we came across. But as you can see from the results here, up to about 15 to 20%, the fold difference between no saliva and saliva is relatively manageable. PCR reactions are even less susceptible to inhibition, as you can see with our SPUD assay. And if you actually use a proper artificial sequence, which is in our test, you can see that up to 20% saliva in the reaction doesn't really affect the efficiency of the assay. Now, this is important because it means that we can extract RNA from saliva samples using fewer washes, which again results in faster extraction times. So this is then our, what we call the Eer workflow. It involves dropping saliva into a tube that contains a modified lysis buffer and vortexing for 10 seconds. Sorry. Also present in that buffer are dynabeads and with biotinylated oligonucleotides with 3' ends, and they target 2 viral and 1 human gene. We incubate this for 5 minutes. We collect the beads at the side of the tube and wash it once, and then resuspend an RT master mix, which contains random primers and RT, and we do 1 minute RT. And so this means that we can subject the sample now to a separate PCR reaction, which means we can have optimal PCR conditions. In addition, of course, the sample now can be transported at room temperature because it's DNA and it is not going to degrade. So we haven't optimized the process yet, so I'll just show you some work in progress. But we are still losing sensitivity, as you can see here. The light blue is the pure RNA. The dark blue is, the dynabead purified DNA. RNA. And you can see we're getting dropouts here. But we have done some tweaking, and you can see this is already better, and this is even better. So I think eventually we're going to end up with a procedure that will maximize the recovery of RNA and will allow us to do all of this manually. At the moment, it takes us 10 minutes. But if you use a microfluidics type system, you should be able to reduce it to less than 5 minutes, we hope. So my second topic then concerns the reliability of the Rtq-Pcr reaction. One of the concerns you hear a lot about is that it is an RNA virus, and so there could be mutations within the either reverse transcription primer or the PCR primers that could influence the specificity or the sensitivity of the assay and give you false negative results. In fact, there is some evidence that that can happen. In fact, there's a whole website that is dedicated to looking at this and identifying mutations and making them visually beautiful. So we decided to look at the whether this was a problem in our particular case, because once we had designed the assays back in February, a few examples of mutations that arose in the forward primer binding site of 2 of our targets have been reported. And so we used our primers on mutated targets. And as you can see, the position at -9, the mutant position at -9 has very little effect on the results that we're getting. We also did it the other way around and made some mutant primer just for the fun of it to see what happens if you use that on the wild type virus. And again, there's some effect, but it's not a major effect. So obviously this is at position -9, and depends where your mutation arises. You could of course get an effect. And the N gene mutation also has some mutations, and again these are the results for the standard wild type primers on the mutant target. And again, by qPCR there isn't much detectable. By digital PCR there's possibly some effect. And if you do the reverse, again, a similar result. So this is of course not to say that just because the results in this particular instance are tolerable, that some mutations will not have deleterious effects. Clearly they can have, and these were all in the forward primer, and you can imagine that in the RT primer there could be more of an effect. However, we conclude that many mutations will actually not make a blindness bit of difference to the reliability of the assay. Nevertheless, we thought it might be sensible to target more than one viral sequence, and so our initial multiplex assay detected 2 on different channels. But then we also thought, well, perhaps we're not really interested in which of the viral targets are being detected. We're just interested in detecting virus. So we could use the same fluorophore for multiple targets, and this might not just make the assay more reliable, but also might increase the sensitivity of the assay. And so these results show, these are qPCR results, that if you have 2 targets, same fluorophore, you get a slight increase in the sensitivity of the assay. And this is confirmed by the digital PCR data, which results in an increase of about 50%. So if using 2 targets, why not use 3 targets? So we tested whether targeting 3 virus sequences has any effect. And so what we did was we replaced the Texas red fluorophore on the N gene with FAM. And again, by qPCR, we found a barely detectable possible increase in sensitivity, which again was kind of confirmed by the digital PCR data. So whether this is ultimately essential to do that or useful is another story, and I'm not saying that this is how it should be done. But what I'm saying is that there certainly is the potential to maximize the reliability and the sensitivity of the assay design by judicious target choice. And certainly I do not think that detecting a single target is sufficient, because I don't think it is reliable enough. So finally, I would like to discuss speed, which has always been one of the big unexploited advantages of the PCR reaction, and where it scores wildly over something like RT-LAMP. Most protocols recommend ludicrously long RT denaturation and polymerization times, and it is easily possible to abridge runtime significantly by reducing these. So you can see here how I've reduced the PCR runtime to on the Bio-Rad CFX, which is not meant to run fast, to 32 minutes, simply by using very, very short activation and denaturation and annealing times of 1 second each, as opposed to 5 and 15, which is also even fast, because some people use, 45 seconds and minute times for this. So I've already told you that we use 1 minute RT times. So combining a 1 minute RT with a very fast PCR allows you to really maximize the speed of the assay. Now, Jim Huggitt and I published a paper a couple of years ago where we showed that reducing denaturation and increasing polymerization times also allows for faster runs on block-based instruments. And the results you can see here show this for the initial sets of assays that I designed. Now, of course, this requires a fast instrument, and we use for this kind of experiment the PCR Max Eco, and we can do the whole run in less than 16 minutes. This is obviously assay dependent, but clearly, as many of you will know, any assay can be tweaked through modifying its design. So by reducing the times and the temperatures, or changing the temperatures, you can really get very fast PCR runs. However, When you're doing this, of course, then plate scanning becomes an issue because it takes 5 or 6 seconds for a plate to be scanned, particularly if you are using more than one fluorophore. So endpoint assays don't have that because by definition they detect the fluorophore at the end of the run. So you can have a much faster one using endpoint detection, but endpoint detection negates the advantages that are inherent in the quantitative aspect of qPCR. So we've modified our real-time detection, and this is what we do. We collect fluorescence data at defined cycles, and the first data point establishes a baseline for that sample. And any increase in fluorescence at subsequent cycles is associated with the likelihood of virus being present. So base fluorescence, and then the earlier an increase is detected, the higher the likelihood of target being present. And so this approach of recording differences in fluorescence helps overcome the complications that are associated with reporting quantification cycles, and we've just termed this multiple cycle fluorescence detection. So detection at cycles 15 to 30 suggests strongly that virus is being detected, and we'd expect that fluorescence to be continuing to be detected in all subsequent cycles. And so what we're doing is, or what we propose doing is providing some kind of a confidence rating from 5 down to 1, where obviously 5 to 2, which is equivalent of roughly cycle 3, there's definitely virus present, and the person definitely has to isolate and no further testing is required. But if we have a confidence level 1, then which is equivalent to an endpoint, then a positive result indicates a possible positive or probable positive, or whatever you want to call it, and then we would recommend a second test a day or 2 later. So as all other UK laboratories, our local hospital reports CQs. And here are the CQs they've recorded on the samples that we have been working with to develop our testing approach. And obviously numbers are very limited, but as you can see, the majority of CQs are below 30, but approximately 13%, I think it's 5 out of the 40, record CQs above 30, and could be interpreted as either positive or negative, depending on how you determine your CQ. And this is shown by this result here, which is a typical qPCR result, where there's a threshold drawn, and you can see the threshold could record something as positive or negative, depending on where you draw it. And in fact, funny enough, this is one of the things that was obvious during the MMR debacle as well, that the threshold determined whether something was positive or negative. So this is the problem with CQs, and I think Mikhail and Jim will be talking more about that. So this is a typical result we would get with our detection system. So it looks very much like a real-time system, except that it's just a snapshot. So the y-axis shows the difference in fluorescence at each cycle compared to the baseline at cycle 8. And in this case, just to for illustrative purposes, I've run 4 replicates for each sample. And you might not do that in a real-life situation. Certainly in our hospital, they do not run replicates. If you then represent the data slightly differently, you can see how you might interpret the data. So for what we call level 5, you get clear amplification at the lowest threshold cycle, which would be a 15. And for cycle 4, there's nothing at 15, but there's something at cycle 20, and so on. Now, again, the precise definition of which cycle you want to use and exactly how you want to do that is up for discussion. Really, all I'm doing today is presenting you with some ideas that we are playing around with, and that might stimulate some discussion. But I do consider that this kind of approach is more reliable and intuitive means of recording the presence of SARS-CoV-2 in a sample. And so this is how we report our result then on a scale of 1 to 5. We also mentioned the fact that there's a mutant present. We've shown that in our case we use c-Jun as our human nucleic acid, and we show a CQ for our artificial sequence because we expect it to be somewhere between 25 and, well, around about 25. It's an artificial sequence, so that's where it always should come up, really. So this is how we would propose presenting the data. So in summary, Testing should be carried out using saliva, in our opinion. The extraction step should be coupled to a target enrichment and reverse transcription step. We should be looking to detect multiple targets, at least 2 targets, and probably in a single channel, which would have the added advantage that you could add other pathogens to this test. And detection should combine the advantages of real-time and endpoint detection to allow more consistent and less subjective reporting of test results. And in conclusion, even the best test, and no matter what we say here, it's all useless without a supporting public health, regulatory and testing infrastructure. So whilst I or anyone in the audience today can make recommendations until we're blue in the face, nothing will change until the various stakeholders coordinate their messages and efforts. And finally, politics always trumps science. And unfortunately, most politicians are ignorant or incompetent, or indeed both. And the scientific establishment from the WHO down has not covered itself in glory either, and the large proportion of the media are blinkered, and they choose to push a political rather than factual agenda. And so I would like to think that our voices and those of numerous other scientists around the world will be heard and incorporated into response to the next pandemic. But I'm not hopeful that this will happen. Thank you very much.
Thank you for that presentation, Dr. Bustin. As a reminder to our audience, you may ask questions at any time using the Q&A tab. Our panel will have a chance to address these questions during our live Q&A session following our 3 presentations. We have a slight adjustment to our schedule this morning. Our next speaker will be Dr. Mikhail Kubista. Dr. Kubista was one of the pioneers developing the qPCR field, inventing light-up probes. In 2001, he set up the Tata Biocenter as a center of excellence in qPCR and gene expression analysis, with locations in Gothenburg, Sweden, and Prague, Czech Republic. The Tata Biocenter is the largest provider of qPCR training globally and Europe's largest provider of qPCR services with ISO 17025 accreditation. In 2019, Tata was named Best Nucleic Acid Analysis Service Provider Europe by Global Health and Pharma. In 2014, Tata introduced non-invasive prenatal testing in Sweden, resulting in the founding of Life Genomics AB. Dr. Kubista co-founded the sports genetic company LifeTest in 2019 and Simsen Diagnostics in 2020 for liquid biopsy analyses. Dr. Kubista co-authored the MyKey and DMyKey guidelines for qPCR and Dpcr analyses, and he is a member of the European Committee for Standardization ISO working group developing guidelines for molecular diagnostics. Most recently, Tata invented two-tailed PCR for the analysis of short nucleic acids and direct blood genotyping. Since 2007, Dr. Kubista also heads the Department of Gene Expression at the Institute of Biotechnology, BioSev, Czech Academy of Sciences. During the pandemic, Dr. Kubista's team was one of the first in Europe to start testing for SARS CoV-2, and the Tata Grand Performance SARS CoV-2 detection kit has been registered for CE-IVD. Tata contributes to the Swedish public testing effort and also operates the platform www.coronapassport.se for travelers and industry. Dr. Kubista, let's make sure your slides are up and running for the audience, and everything appears to be fine. So, Dr. Kubista, the floor is yours.
Thank you, Nathan, for the introduction. It's a pleasure to be here and share our experiences with you on the behalf of the scientist. So I'm from the Tata Biocenter, which is a center based in Sweden. And I think important is that we are not a diagnostic laboratory. We are actually best known for offering training in molecular analysis to industry, hospitals, and private laboratories. And we work in close collaboration with solution providers around the world. We also offer molecular analysis services to industry, diagnostic laboratories, routine laboratories, and universities. We have an R&D division that develops methods and solutions to various challenges in molecular analysis. Many of the methods we developed are on the market today, usually through partnerships with larger companies. So, what have we done at Tata so far during the pandemic? Well, already in January, we were approached by one of our customers, St. James Hospital in Malta. They were requesting our support to provide a qPCR test for SARS CoV-2 coronavirus. And already 1st of February, we had a test that had been validated in-house, and we started to provide it to to Malta, and the hospital, St. James, started to test. And we also could start preparing for testing in Sweden. At our site in Prague at the Biosef campus, we could support the researchers to set up COVID testing and contributing to the Czech public effort. We also teamed up with the Danish Technical University, Statens Serum Institute in Denmark, Sun Yat-sen University in Jiangsu, China, the Chinese Center for Disease Control, the University Vita Salute and Icons in Italy, and the Medical University in Vienna. And we were fortunate to receive an Horizon 2020 grant to start developing rapid COVID-19 testing. We were also joined a consortium coordinated by the Cambridge University to support COVID-19 diagnostics in Africa. And as mentioned in the introduction, we have validated our SARS-CoV-2 test, registering it for CIVD, and we have also filed for FDA approval. And in Sweden, we support the public SARS-CoV-2 testing effort in collaboration with Sahlgrenska University Hospital, and we also operate the platform www.coronapassport.se, offering testing service with health certificates to travelers. So, let's look into the diagnostics. When assessing a molecular diagnostic test, most critical is the diagnostic accuracy. This is essentially a characterization of how well the molecular test correlates with the clinical situation. Most important parameters are the diagnostic sensitivity, which is the fraction of disease cases that test positive, and the diagnostic specificity, which is the fraction of healthy subjects that test negative. In a normal situation, when the disease and clinical picture are well established and the diagnostic method fully validated, these parameters are most critical. However, when a disease is new, it may not be trivial to make clinical diagnosis, to separate the new disease from related diseases based on symptoms only. There is also no reliable independent diagnostic method available to compare with. Furthermore, the assessment of the accuracy depends on the population tested. For example, asymptomatic individuals may test positive with a molecular method targeting the virus before any clinical symptoms appear, and correlation will be much higher when testing patients in, for example, intensive care units. So, most validation protocols on diagnostics, they do require some assessment of diagnostic accuracy, including those for SARS-CoV-2. Even us collected these kind of data for our registration, as it was required. But at early stage of a new virus appearing, those data are not particularly informative, and they are not comparable across studies, and they are not suitable for comparing of tests. We and many others are working with qPCR. Analyzing with qPCR, we add primers that amplify the targets, and if it is present, it gives rise to an increase in fluorescence as the number of amplification cycles increase. This is a typical qPCR response curve. So how do we quantify the amount of targets, such as viral RNA, using qPCR? To approach this, let's consider 3 samples rather than an isolated response curve. And we introduce a threshold line, which is set essentially as a horizontal line at an arbitrary level. And we read out the crossing point between the amplification curve and the threshold line. Those crossing points, the fractional cycle number, is referred to as the CQ value according to the MICI guidelines, although many diagnostic laboratories still refer to it as CT. Nevertheless, CQ and CT refer to the same thing. We can now compare the samples pairwise, calculating the difference between their CQ values. The difference is referred to as delta CQ and is seen in the graph. The delta CQ reflects the distance between 2 qPCR response curve at the height of the threshold line. Recall, the threshold was set arbitrarily. So what does happen if we change the threshold? As seen, if we raise the threshold, all CQs are shifted towards higher values. Clearly, the CQ values depend on the arbitrary threshold. But what about the delta CQs? As seen, they remain the same. Hence, we conclude that absolute concentration measurements with qPCR are not really possible. Only relative measurements are. Many routine diagnostic laboratories report CT or CQ values by tradition. Well, under fully standardized conditions, where only one instrument is used, operated with the same protocol, targeting the same target, and always analyzing samples processed with the same workflow, the data may be comparable, although not necessarily possible to convert to absolute concentrations. However, in a situation where different protocols and instruments are being used, the measured CQ values depend on multiple factors and are not comparable. Those factors include the instrument, readout procedure, the particular algorithm used by the instrument, the reaction volume, the particular master mix, the primers and probe sequences and their concentrations, the reaction conditions, and containers, to name just a few factors. So, how should one really assess a test's performance? We will illustrate this with an example. The valid prime is a highly optimized assay targeting human genomic DNA. We will now assess the assay's performance using standard material calibrated against the NIST human DNA quantification standard. And just in case, the concentration of the standard material was further verified using digital PCR. For convenience, the assessment is performed using the high-throughput IntelliCube qPCR instrument from Douglas Scientific. So, when assessing a qPCR test, a static curve is performed with a concentrated stock solution of known DNA concentration that is sequentially diluted by a factor of 2. At each concentration, we performed 64 or even 128 replicates. This is a residuals plot. This is essentially the standard curve tilted into a horizontal position. inspecting the data, we see the spread of replicates increases towards lower concentration. This is expected due to sampling noise. Sampling noise is observed when there are few target molecules. Let's assume a patient infected with a virus having a titer of 1 viral molecule per milliliter of blood. If we sample 1 milliliter, that particular volume may contain a single molecule, but it may also accidentally contain 2 or perhaps even 3 or 4, and it may be 0. The probability it contains a particular number of RNA molecules is given by the Poisson distribution, which is plotted here for a few selected cases. The dark green curve is the case when the viral load is 1 molecule in average per mil. From the graph, we read out the probability is only 37% to sample exactly 1 molecule. It is 28% probability to sample 2 molecules and about 18% to sample 3. Using the Poisson distribution, we can calculate the irreproducibility of measurements due to too low numbers of target molecules. This is shown here for the standard deviation of the measured CQ value of replicates in a qPCR experiment. This is indicated on the y-axis. As seen, the standard deviation increases when the number of target molecules indicated on the x-axis decreases. The green line is drawn at a standard deviation of 0.25 cycles, which is the specified repeatability of most qPCR instruments by the manufacturer. As seen, an SD of 0.25 cycles is also introduced when analyzing samples having 35 target molecules in average. Hence, when quantifying samples with less than 35 target molecules in average, the confounding variation due to sampling noise becomes larger than the noise introduced by the qPCR instrument. A diagnostic test for quantification is typically characterized by its limit of quantification, also referred to as LOQ. LOQ is defined as the lowest amount of target in a sample that can be quantified with stated acceptable precision and stated acceptable accuracy under stated experimental conditions. Hence, we need to agree on the acceptable precision and accuracy. This depends on the purpose of the test and cannot be decided by the laboratory. Rather, it should be decided or agreed with a clinician who specifies the accuracy needed for making clinical decisions. This depends on the disease and the clinical situation. Still, quite common is an imprecision of 35% in linear scale. That is, we accept an error of 35% in number of molecules. A coefficient of variation of 35% in linear scale corresponds to a standard deviation of 0.49 cycles in log base 2 scale, which is the scale of CQ values. So, entering the graph we had before that is constructed by the Poisson distribution, we see that a standard deviation of 0.49 cycles corresponds to an average concentration of 10 molecules. Hence, the noise when analyzing samples containing 10 target molecules in average introduces an uncertainty that is around 35% in linear scale. Hence, with this restriction, we cannot quantify samples containing less than 10 target molecules. Importantly, note this limit is so what about real tests? Well, here are the data from the valid prime standard curve. The standard deviation was calculated at each concentration from the replicates and recalculated to the relative error in linear scale. The red dashed line is at 35% relative error. So we can read out. We can read out the limit of quantification for the valid prime assay as being between 16 molecules, which is the lowest concentration with a CV below 35%, and 8 molecules. The limit of detection, LOD, which sometimes is also referred to as the analytical sensitivity, is the lowest amount of target in a sample that can be detected with stated probability, although perhaps not quantified. Working at 95% confidence, this corresponds to a target concentration when 95% of replicates are positive. Hence, if a test at LOD is replicated 100 times, 95 are expected to be positive, while 5 are expected to be negative. This graph is also constructed from the Poisson distribution and shows the fraction of replicates expected to be positive under ideal conditions as function of the average number of target molecules per reaction volume. Entering the graph at 95%, we read out that the theoretical limit of detection is 3 molecules when the test is suffering from sampling noise, which in practice are all diagnostic tests, as we never analyze the entire subject. Hence, irrespectively of technique, we cannot reproducibly detect less than 3 target molecules. So here are the experimental data for the valid prime assay. The fraction of positive replicates is plotted as function of target concentration and is fitted to a sigmoidal function shown in blue. The green line is at 95% positivity, which is the confidence level we work at. It crosses the blue line at 2.5, which is the experimentally estimated limit of detection for the valid prime assay. The red solid lines indicate the 95% confidence area around the blue line, and from their crossing points with the green line, we read out the 95% confidence of the estimated limit of detection. Hence, the experimentally estimated limit of detection of the valid prime assay is in between 2 and 3.7 molecules. This plot shows the original valid prime standard curve, but now limited to the quantifiable concentration range, that is, above limit of quantification. Fitting the data to a straight line, we estimate the PCR efficiency to 99.6%, with a 95% confidence interval ranging from 97.8 to 101.4%. Summarizing the data, we show that if you have a highly optimized assay, you indeed reaches theoretical limit of detection and limit of quantification. The approaches are published, and here's the reference to the paper. Still, what about the pre-analytics? Characterizing the PCR or for RNA targets, the Rt-Pcr is nice, but a lot is happening to the sample upstream of the PCR that influences sensitivity and reproducibility. The sampling is, for example, is expected to introduce variation due to the operator. Compare a skilled medical professional with a sick person doing home testing, and you will see. Then, of course, the tested body part. Is it a nasopharyngeal swab or is it saliva? The swab material and the procedure are also expected to introduce substantial variation. The transport medium is essential. Some swabs are transported dry, others in various buffers that keep the virus intact, and still some in chaotropic media that kill the virus. The volume of the medium, of course, matters as well. And then how the swab is extracted to transfer the virus into the medium. RNA is not a particularly stable molecule. In fact, when the virion is opened, releasing the RNA in most media, the RNA is degraded over time. Degradation rate depends on the medium, but also on the temperature and light exposure. And before the Rt-PCR, the RNA must be extracted and purified to be accessible and to minimize inhibition. Many extraction methods and protocols are available, differing in critical parameters such as input and output volumes, to name just a few. And then we have the fraction of the extracted material that is used for the Rtq-Pcr as another variable. So, experimentally determined or estimated LOD and LOQ reflects the analytical workflow only from the point the calibrated standard is added. Performance upstream of that point remains unknown. Most SARS-CoV-2 methods available are only calibrated from the extraction step, and some even only for the qPCR step. Hence, there is no control of the sampling, handling, and transportation. These pre-analytical steps are expected to be most confounding. To optimize an entire workflow and assess its performance, that is a costly and tedious process that takes time. It is illustrated here. The workflow should be broken down into steps, and replicates shall be introduced in between these steps. This is known as a nested experimental design, and statistics can then be applied to estimate the confounding variation introduced by each of the experimental steps. This allows for optimization and also assess the overall performance of the entire workflow. This has not been done yet for any SARS-CoV-2 Rtq-Pcr-based workflow. So what is reasonable to do? Like most others, we at Tata have calibrated only the extraction and the Rt-PCR step. We have also used different positive controls. In the early days of the pandemic, the only positive control available was rather short synthetic RNA fragments. Then in vitro transcribed SARS-CoV-2 RNA became available, and then also RNA extracted from SARS-CoV-2 infected cell lines. And most recently, also encapsulated RNA is available. The latter mimics the virion and can be used to assess also the transport and storage conditions. No matter the RNA standard used, the concentrations stated by the manufacturer must be verified. Our experience is the stated concentrations can be way off. Easiest is to calibrate by determining the concentration with digital PCR, as is shown here. This is a validation of our Rtq-Pcr workflow. A standard curve was performed covering the range from about 50,000 to 5 target molecules per reaction, establishing the dynamic range. Separately, limit of detection was estimated using 20 replicates of 10 copies per reaction, shown in bottom left, and 5 copies per reaction, shown in bottom right. And here we validate the extraction and Rtq-Pcr steps combined. Left is a standard curve between 10,000 molecules and 1 copy per qPCR, and to the right are 20 replicates at 10 copies per reaction, shown in red, and at an average of 3.3 copies per reaction, shown in green. For the low concentrated replicates, about half are positive and half are negative. So 3.3 in this setup is a little bit below the limit of detection. And this is an example of a run. 96 samples are analyzed in our routine pipeline. 1 positive control is included, which here has a CQ around 27. One of the test samples is positive, which has, in this case, a CQ around 31. The rest of the samples are negative. Note that the negative samples never amplify, and therefore they don't have a CQ value. So what kind of lessons have we learned, and what can we share? This is, of course, very difficult, but at least some thoughts for the laboratories. Number 1 is don't compare CQ values. When everybody uses a different workflow, it just causes confusion. Simple positive standard material were early made available by oligo houses. Use it to provide an estimate of what is being detected. It's valuable, at least for internal use. Importantly, always provide information about the fraction of the original sample that is analyzed. When comparing laboratories for whatever reason, like performing a tech transfer or proficiency testing, make sure target concentrations are above limit of detection for qualitative analysis and above limit of quantification for quantitative analysis. If concentrations are too low, the comparison will fail due to sampling ambiguity, and it does not reflect the performance of the laboratory. And when comparing samples in different laboratories, make sure they are treated exactly the same way. That includes storage and transport conditions. Remember, RNA degrades with time. For the kit manufacturers, correlation with disease is, of course, essential. But as I said in the beginning, be aware of the shortcoming in early stages when a new pathogen appears. We may not know or understand the clinical symptoms it causes in the beginning. Validation for cross-reactivity to other pathogens and species is essential. In an emergency, this can be done quite reliably in silico. Specificity for the targeted species is also essential, as mentioned by Dr. Bustin. This must be updated as sequencing data for mutations become available. Particularly in the beginning, validation with alternative targets, such as a second or third target gene, can be important. Establish PCR, Rt-PCR performance, including LOD, and when relevant, limit of quantification, and of course, PCR efficiency. Establish there is no qPCR response for negative controls. Establish quality control of reagents, testing every batch for contamination. Manufacturing positive controls at the same site as primers and probes is, of course, not a good idea. Providing positive and negative controls, including verification protocols to customers, is important. And establishing instructions for use for the entire workflow, starting from the sample collection to minimize risk for carryover, inhibitions, cross-contamination, and the like. Regulatory bodies, this is of course toughest. That's not really our field, but we have learned a bit from our side. Knowing the competence in your country is essential, because then we can start fighting a new pandemic earlier. Service providers and core facilities have usually better readiness to respond to an acute situation than routine laboratories. Generally, we can't collect all the data to obtain standard CIVD approval. Too much is required, and we don't have all that information about the new pathogen. So there must be a fast track, like the emergency use authorization that most countries have. They should be quite generous in the beginning, and they can become more stringent as we learn more about the pathogen and the disease. Then the more stringent criteria shall be applied retrospectively on the tests already on the market. The emergency use authorization, particularly in Europe, is not very powerful, and the reason is it's typically very limited, often limited to a particular hospital. The emergency use authorizations should be at least national. Why not European? It's important to have organized information sharing and also qualify that information. Organizations like the WHO and ISO could perhaps be behind that. Training, particularly for all the new labs that must set up a new test, perhaps with limited experience. I mean, the big hospital labs, they know, of course, what they are doing. They are experts. But in a situation like this, a large number of labs are setting up COVID testing with very little experience. It's very easy to arrange training, even on-site training for those labs to help them getting started. Then it's important to identify bottlenecks in the workflow. For example, with the SARS CoV-2, if we would have focused early on inactivating the virus at sampling, then we would not need to be limited to laboratories having biosafety level 2 and 3 facilities, and we could much faster start mass testing. Regulatory bodies, usually their main job is to control the products and the services on the market. And this is, of course, essential. But during the pandemic, it is important they support the laboratories setting up testing. We have been working with regulatory bodies in a large number of countries. And in one country, we were met by the regulators telling us more or less, hey guys, we are in this together. We are a team. We'll get you going fast. We love that. Much of what I have shared with you today is available in guidelines. These are the ISO guidelines for qPCR and Dpcr. These have just been made available free of charge last week as a part of the Corona package. So I suggest take a look at those. And with this, I'd like to thank you for your attention, and I'll be happy to answer the questions, but I guess it will be after Jim's talk.
Thank you, Dr. Kubista, for that presentation. And as Dr. Kubista mentioned, I would just like to remind our audience again that you may ask questions at any time during any of our presentations using the Q&A tab, but our panel will address these during our live Q&A session following our presentations. And our third presentation today is Dr. Jim F Huggitt. Dr. Jim Huggitt studied genetics at Liverpool University and earned his PhD at Cardiff University, where he studied transcription in bone disease. His interest in the application of advanced molecular methods to clinical scenarios brought him to University College London in 2002 to take up a senior research fellow position with Professor Almuddin Zumla. At UCL, Dr. Huggitt investigated diagnostic approaches to infectious diseases in developing world settings. He moved to the National Measurement Laboratory, NML, at LGC in 2009, while still retaining an honorary lectureship at UCL, which he still holds. At the NML, the UK's designated National Measurement Institute role for chemical and bioanalytical measurement, he has led a large number of molecular diagnostics, genomics, and nucleic acid research projects. This work focuses on high accuracy measurement, as well as strengthening the traceability of measurements that underpin legislation, regulation, and standardization. In 2016, Dr. Huggitt joined the University of Surrey as a senior lecturer in analytical microbiology and holds a joint appointment with the NML. Dr. Huggitt, let's make sure your slides are up and running for the audience. And everything appears to be all right there. So, Dr. Huggitt, the floor is yours.
Thank you very much. Can you all hear me okay?
Yes, we can.
Excellent. That's a start. I do apologize for rearranging the schedule today. I've had a complete nightmare with the computer and I've had a rather stressful hour trying to become a computer expert, which I'm certainly not. And you are presenting the PDF slides, is that correct? That I can see here?
That's correct, Dr. Huggitt.
Thank you very much. Okay, so good day, everybody. Thank you very much for being here, and I'd like to thank the scientists for giving me the opportunity to speak to you today. My name's Jim Huggitt, and as you just heard, I work in the UK as a principal scientist in National Measurement Laboratory and senior lecturer. If we could go to the next slide, please. So my presentation is loosely split into three sections. I'm going to give you a quick introduction on the National Measurement Laboratory and a single slide on metrology. And then I'm going to talk about SARS CoV-2 standards and validation and some of the things that have been going on. And then I guess the second half of the presentation is really going to focus on measurement science and how I think, and hopefully you will agree with me after this, it can be useful in approaching and responding to a SARS CoV-2 pandemic, but also other areas as well. If we go to the next slide, please. So this is the DNA Measurement Laboratory at the National Measurement Laboratory, and it is based at LGC, and that's in Teddington, which is southwest London. And we are a designated institute to conduct bioanalysis and chemical analysis. And this is the molecular lab, and you can see it's fairly new, as updated last year and opened last year. So I'm very fortunate to work in this cutting-edge advanced laboratory. Can I have the next slide, please? Our group works on pretty much anything you can think of that might involve nucleic acid measurements and their modifications, DNA, RNA, methylation, and a wide range of sectors, although I think it's fair to say the vast majority of the group has been working on a particular virus over the last 6 months. Can I have the next slide, please? We work, one of the wonderful things about working in the measurement space in the National Measurement Laboratories is it has to be by its very nature international. And so we perform lots of collaborations, and these are EU-funded projects you can see, and some other collaborators wider than the European Union that we've worked on. And the next slide, please, will show you the current study we have, which is called SEPTIMET, which is called Metrology to Enable Rapid and Accurate Clinical Measurements in Acute Management of Sepsis. This started last autumn, or in October of last year, and was timely because many of the challenges we are really looking at in sepsis are also useful in applying some of the approaches to look at SARS-CoV-2. And so we've been working as one of the models on SEPTIMET is now also SARS-CoV-2. Metrology, if we could have the next slide, please. What is metrology? Well, metrology is essentially the science of measurement. We take measurements for granted, whether you're getting in your car, whether you're weighing flour to cook some bread, generally every day, we just make measurements. these do not happen by magic. They are the entire infrastructure globally that supports everyday measurements, ensuring that they are traceable. And when you make the measurement, you are comfortable that you are making the correct measurement. Historically, this has underpinned trade to ensure you have fair, when you're purchasing items, you are being treated fairly, or you are containing what you think they have. But increasingly, the measurements science field has been focusing on clinical chemistry to improve the measurements that occur in everyday medicine. And molecular measurement over the last 20 or so years has really advanced in this space. And it allows us to understand sources of error, both random and biased sources of error. It enables traceability, so you and I are able to make the same measurement and be comfortable we're measuring to the same level, which ultimately improves measurement accuracy. Today, I'm going to focus on analytical performance of SARS-CoV-2 molecular diagnosis. I've underlined the analytical bit to remind me to point out to you that I'm not a clinician and I'm not a virologist. So some of the conclusions I will present are in that space, and how they can be applied afterwards, it's down to those who are experts in those fields. But I just want to make sure that that is what I'm talking about, the actual methodology, not how it may be used in a clinical context. Could I have the next slide, please? And the next one, sorry. So apologies for this having to go with the PDF. So I will now be focusing on, if we go to the next slide now, on the standards bit and the validation. So if we can move to the next slide. This essentially is the method, and Professor Bustin and Kubista have really just spoken about this at length. But this method, reverse transcription real-time quantitative PCR, has essentially made it possible for us to respond to the SARS-CoV-2 outbreak and the COVID-19 pandemic. I wonder how and where we would be if we didn't have PCR in this case, and it is quite frightening to think in that context. But essentially, the PCR plot, as you see here, is conducted over cycles. You run up to 40, maybe more cycles of PCR, and you monitor the fluorescence during the experiment in real time. And depending on how you set up the experiment, you're able to you measure the increase in the PCR product with fluorescence. And in the blue line and the red line you see there, essentially the blue line comes up earlier. It is coming up. It is amplifying at an earlier cycle than the red line, because we assume there is more nucleic acid at the beginning. Now there's an entire afternoon or a couple of days with discussions around what can influence that. But that's essentially a lot of the discussion we have is around that. However, if we go to the next slide, and there were animations here, but I apologize, we can't have them because I'm having to use the PDF. SARS-CoV-2 testing is more complicated than the analysis only. And Mikhail Kubista has really shown this clearly. Professor Bustin may have done as well. I apologize, Stephen. I was trying to sort out my computer, so I didn't see a lot of your talk. But essentially, the analysis bit, the detection of the viral genome, is only a small component. We have to decide on the specimen, and Mikhail showed the storage and the sampling, but there's also the processing to purify the viral genome. And all of these steps add potentially, they will add uncertainty, error. They can add both variation on the measurement, whether that's quantitative or otherwise, but they can also add bias. An extraction that is not very good will not purify all the virus. And so that can impact on the quantities you're measuring, and ultimately could impact on whether you measure the thing or not. And the specimen choice is we know now, or we've known before actually, is widely variable. Nasopharyngeal swabs some take, they can be easily taken. In the United Kingdom, you can go to a laboratory or somewhere where someone who knows what they're doing, an expert clinical healthcare person will take the sample. But we're also doing home sampling, so you can guarantee that there's going to be a difference in the quality of that sample, and that will impact how much RNA ultimately gets into the experiment, the PCR for analysis, and which will also impact on the technological performance. If I go to the next slide, please. When the SARS-CoV-2 outbreak started at the beginning of the year, around, one thing that occurred fairly fast was that the WHO published some primers sequences for people to use, and these were made available, and people used them. And there were a range of different ones. And what was clear and what we can potentially learn as a community is that there was a spectrum of the way these were presented. Some had internal positive controls as well. Some had information on how to run them. Some were literally sequences. And so this is a potential area when we're moving to the new pandemic to perhaps think about a strategy for providing information on how people can apply these. Because taking a set of primers and running them with some control template is not that difficult. What can be challenging, though, is to get to know how well they're working, to know when and how they should be working, what to expect. If we move to the next slide, this has been somewhat complemented by the fact that now there are large numbers of commercial molecular diagnostic tests. If you go to the Find Diagnostic website, which you cannot see because my animation is not working, but if you type Find Diagnostics into Google, you will find them. There are now 366 commercial tests available, some of which have regulation associated with them. But the trouble with some of these tests, and I'm not going to name any names, is that everybody appears to be a SARS-CoV-2 diagnostic provider now. No matter what you did before, you could be focusing on a completely strange genetic scenario which requires epigenetic measurements, but you're offering a solution to SARS-CoV-2 testing. And so this also potentially provides some, if you're a person wanting to set this test up and you're not sure which one to go for, having 366 options does not really help. What you would also see if you go to this website, that I cannot show you again because the animation is not working, is that they, in some cases, they have conducted some analysis and evaluated some of the performance of many of these methods. What they find with some of the PCR-based methods is that they do not necessarily measure low signal. They don't measure tens of copies or sub-10 copies of template. Now, to my mind, PCR, I'm not talking about isothermal methods, but a PCR-based approach should be able to measure single copies of, or near Poisson, as Mikhail showed, in the PCR. I'm not talking about the extraction, I'm not talking about what was in there before, but if you're taking purified nucleic acid, this should be an expectation, I think, of an assay, that it should be able to measure pretty low signal. Because again, when you're turning to the actual sample proper, the bar is set by hundreds of copies per mil of sample. And so that is an important factor, I think. Next slide, please. From the point of view of standardization, the communities involved, the National Measurement Institutes and the standard providers have been busy. And we have on some publications, both from our group and from NIBSC, looking at the need for standards. The CCQM is, I will touch on a little bit in a minute, is the consultative committee for the measurement of things, essentially, chemical and biological entities. And the Nucleic Acid Working Group is working on SARS-CoV-2, as I'll touch on. And the Coronavirus Standards Working Group, which is being, I guess, it's being fronted by Mark Sallet of the Joint Initiative for Metrology and Biology in Stanford, is also doing lots of activities trying to support global standardization. An example, if you go to the next slide, please, is this database which was put together by people, well, Alexander Whale, Gerwin Jones from NML and Megan Cleveland from NIST, whereby you can go to the database and select your control material, and you can choose a variety of different types of materials. And what's good about this is at the top you can see it tells you what stage in that process, from sample taking, transport, lysis, purification, reverse transcription, and PCR, the control material is expected to control for, because that's not necessarily apparent. And as you can see, the ones here we have ones that are selected that will do lysis. They are viruses or viral-like particles. And in the middle at the front there, you've got JLC and Twist as an example, which are they're purified nucleic acids, and these will allow you to rigorously evaluate the reverse transcription and the PCR to try and get that to work as well as you can. Next slide, please. And I think this has gone the wrong way around. Apologize. Next slide, please. Considerations, though, that we've found in working in this space is which will lead to discrepancies before you even go near your PCR machine, are the use of some of the units. Some of the materials are in copies, some of them are in nanograms. Some of them are other base mass strategies. The denominator is the key thing that makes me smile, though, because some are per mil, some are per microliter, some are per pellet, some are per tube. So immediately before you've actually bought the thing, you have a discrepancy in the traceability because you're not entirely sure, and you've got to think, well, okay, how do I compare that one with that one? Now, you may decide you don't need to because you've chosen a particular source. But when you want to compare your work with somebody else and they've chosen a different source, it can be a bit challenging to work out how copies per mil might link to nanograms per pellet. The format that they come in is important to realize as well, and the fact that if you are having, if you are trying to optimize the PCR, you may not want to have a whole material because you need to extract it. However, if you want to evaluate the whole process, a buffered solution, an RNA molecule that is purified, may not be suitable. And we have the terminology that's used. These materials are quality control materials, they're standard materials, sometimes they're called reference materials. And these terms tend to be interchangeable, whereas there are clearly defined guidelines in ISO as to what certainly a reference material should be. And so we need to be cautious about perhaps using some of those terminologies. The quality of them as well, and I don't mean the quality as in, are they any good or not? I mean the composition and the things around those. So for example, if you are going to purchase a purified in vitro transcribed fragment of RNA to optimize your PCR, that you might have some idea that is a pure molecule. It's just the molecule you think it is. However, if you're going to obtain some cultured virus, that may not have the same representation of different positions of the genome. You may find that certain parts are more abundant than others for a variety of reasons. And you may find there's fragmentation of some of these materials. And this is not a bad thing, it's knowledge, but it may mean that if you are using a method that detects a 300 base region of the genome, as opposed to one that detects 60 in case of some qPCRs, you might find you get different results simply because of the composition of the material. And so we need to be cautious with that. Next slide, please. A good example is value assignment. This, and I'll touch on this in a second. The SARS-CoV-2 diagnosis of infection, whether that's linked to disease or not is to be decided, is pressing that it's presence or absence at the moment. There's discussions around quantification, which I will touch on the end, but at the moment it is presence or absence. And many of the materials that you will be able to access are clear that they are nominal values. The values they have made are a guide. But many of these materials, though, are used to provide CE marking, and so you're finding, or FDA approved, and so things like limit of detection are turning to the quantities put on these materials that are not necessarily, as the vendors of the materials will state, have necessary for that role. So you'll have situations like this where you're measuring almost homeopathic amounts of virus because the calibration is not quite right, because the materials have not been rigorously characterized, primarily because they were not intended to be. And so if we move to the next slide, here's an example of a QC material we received, and I'm not going to name them because it's not intended. Essentially, the values are supposed to be at 1 million copies, and we are measuring a variety of different levels with different lots. But we don't know if that's the lots. We don't know whether the people who've sent us the materials have handled them correctly, how many times they've freeze-thawed them. There are all sorts of issues involved in measurement that you would do if you're going to apply these methods to do limit of detection or start to quantify, that we must factor in. Homogeneity of the material. Many of these materials are not designed for that, and so it's not fair to expect that they can be used in that way. Can we have the next slide, please? However, routine testing requires quality, and it, has to be so. One of the most amazing things about the last 6 months is how some of the proficiency testing schemes and the external quality assessment providers have really stepped up to the mark to provide materials that provide the world with confidence. And this is happening now, that they're providing diagnostic manufacturers and clinical testers the confidence that their methods are working. And again, the clinical diagnostic providers, they need to know, and they need this level of support. And really the proficiency testing scheme and the EQA schemes have done a fantastic job over the last 6 months in meeting and providing that level of support. Next slide, please. However, this is a quote from some from dialogues I've had where people who've participated in it are perplexed that their quantification is different from the PT schemes. But the reality is that the PT scheme is not designed to quantify. The values are a guide, but they're not supposed to be used for quantitative performance. But the qPCR, we're using the qPCR because it's automated. You don't have to, you do your experiment, it's real time, you run it. But it gives you a quantitative value whether you ask for it, or it gives you a CQ value whether you ask for it or not. You may not choose to look at it, but people are. People are tempted to look at it. People are trying to use this as some kind of metric, and I'll touch on that in a minute. Next slide, please. So just to kind of conclude this bit, today there are numerous quality control materials available, a wide variety of different applications, and many assays, both commercial and non-commercial, that you can choose from. But traceability challenges make it difficult to know how to compare the methodologies at the analytical level, and so that's going to have an effect on the clinical measurement and their performance. Things like the quantification, metrics like limit of detection, which are important components in a diagnostic test. But the systems are in place to support non-quantitative tests at the moment, and generally the laboratories are performing well. And I have that in red and italicized because the labs are being supported by the proficiency schemes, and they are able to meet what the presence. And generally, from what we understand, is they are doing very well. The question would be, and it would be a debate with the proficiency schemes, as to what level the laboratories are really being challenged, and B, what level do they need to be challenged in terms of abundance and the amount of materials that this should be measured in the context of the type of diagnostic they're delivering. And this is an ongoing conversation, and I certainly don't have answers at this stage. If I would ever have in what we do anyway. Can I move to the next slide now, please? Now we're going to talk about what can measurement science tell us. So if we can just move to the next slide, and the next slide. Would have been easier if I'd had the animation. So measurement science. So there are a large number of, well, probably a small number actually. There's a number of National Measurement Institute laboratories across the world who are fundamentally interested in the performance and measurement of variety of things, and DNA is in that category. And so here's an example of some work that was led by Chris Korea, the Korean National Measurement Institute. Some absolutely amazing work that essentially demonstrated that we have potentially got SI traceable measurement of DNA. And if you move to the next slide, one of the methods that's allowing us to do that is this digital PCR that both Professor Bustin and Mikhail Kubista have already touched on. We're interested in digital PCR in the National Measurement Institute community because it counts the DNA. And if it counts the DNA with a level of accuracy, and if we know that we will look at defining, and we know that the volume can be well defined, then we have potentially a method to measure all of the DNA molecules with SI traceability. You have the counting is the one stream, and then you have the volume, which is the other. And that allows us to potentially measure with very, very high accuracy. And we know that digital PCR is very reproducible, but we have to prove that. So if you move to the next slide, this was some key work, and unfortunately you can't see the paper, publication unfortunately, but it was in clinical chemistry, and where we were looking at KRAS. And essentially you can just about make out the figure on the right-hand side. You can't see the x-axis, y-axis, this doesn't really help, but the digital PCR technology is highly reproducible. And we believe it's close to SI, certainly when measuring DNA like KRAS. And that means that I can give you a piece of DNA molecule, say a plasmid, and you can measure it with the digital PCR, and you're likely to get very similar results to me without worrying about calibration. This is a major problem with qPCR. There's just no way to do that. We have to worry about calibrators. And so this basically demonstrated that this is possible. If we move to the next slide, please, possible for DNA. But we're looking at RNA for SARS-CoV-2. Well, the CCQM, which is the nucleic acid working group of the CCQM, which is one of the groups that essentially arranges studies and key comparisons for national measurement institutes to demonstrate their competency, has conducted a number of RNA studies, mainly looking at messenger RNA to begin with. But last year, P199 fortuitously worked specifically on HIV RNA. And which put us in a very good position to fast-track the study P199B, which is currently happening, which is looking at SARS-CoV-2 copy number quantification in collaboration with probably the biggest study or biggest number of participants we've had. I think it's about 21 laboratories, and that is led by the National Measurement Laboratory, but is co-organized with National Institute of Measurement in China, the NIST, National Institute of Standards and Technology in the USA, and the National Institute of Biological Standards and Controls, NIBSC, in the UK. And the results from that will hopefully be out towards the end of the year. Go to the next slide, please. Why should this matter for SARS-CoV-2 diagnosis? Arguments that I've been in some conversations with in the UK where fairly eminent decision makers have said, this is a simple process. We don't need to worry about high-level measurement. It's not really an issue. The virus is present at 1 million copies. The PCR can measure 1 copy, and we don't really care about how much is present. So it shouldn't really be a problem. Well, those are valid arguments under certain circumstances. If we can move to the next slide, please. Well, here's an example of where quantification is used to compare, in this preprint, different samples. And so that you can see from this that the argument that the virus is present at 10 to the 6, 1 million copies per mil, is valid. But those of you who are a bit, perhaps more interested in the statistics side, will also see that there's quite a big distribution on that, which got us interested because those arguments are valid for certain circumstances, but it largely depends on the distribution of the data. So we can move to the next slide. And I need to get this up now. I really have to thank two, and actually there's a third group coming in on this, but I'm unable to show their data. Great Ormond Street Hospital, Catherine Harris and Julian Brown and their team have been fantastic support. We work very closely with them. And they were able to provide 607 positive results. And then Jo van der Sompelen from BioGazelle was then able to provide 4,000 plus results from the BioGazelle as part of the Belgian SARS-CoV-2 testing platform. So I'm now going to show you their data, and I'm going to discuss in the context of where 1 million copies, when we can assume that is okay. If we move to the next slide, here's an example of where 1 million copies is and just saying, look, don't worry about it, everybody. 1 million copies is probably enough. Yeah, that's fine. And this is a completely hypothetical scenario I put here. What you have is, and this is based on the Great Ormond Street data, their qPCRs, you have about 100 copies is about cycle 40, which corresponds to about 1 copy on the PCR, because 100 copies per mil has to be extracted, processed, and how much you actually get into the PCR is limited by physics. And so about 100 copies is the approximate limit of detection of the Great Ormond Street assay, which means if you do the maths, at about 25, 24, 26 is about 10 to the 6 copies per mil. If this was the distribution, then actually, having measured that, the measurement scientists say, well, actually, it's fine, everybody. You're probably going to be okay, because while you have almost 10 cycles CQ spread in the data, you've got here the frequency on the left, and you've got the PCR cycles on the x-axis, and the y-axis you've got frequency, this histogram. You can see that the vast majority is peaking at about the 1 million copies, and we have a distribution, and that's 1,000-fold across there. 1,000-fold, that's quite a big distribution if you think of other clinical chemistry measurements, but you have, importantly, quite a lot of space, and no noise, no signal between cycle 30 and cycle 40. And so there's no low signal to measure, and therefore, actually, it should be fine, shouldn't it? Because don't worry, we just measure it. And many tests, many clinical chemistry tests work on that basis, the fact that you're measuring a large amount and there's not a big distribution, and there's a big difference between the limit of detection and the actual range that's being measured. So we move to the next slide. We now think about, well, we now plot the Great Ormond Street data. And if we could go to the next slide, you can see that this is not the case. 1 million copies is certainly there. If we move to the next slide so I can get rid of the hypothetical data set you see that there are some very low signals, 16 signals. There's a few patients that have very high viral titers. It starts to increase about cycle 22, 24, 26, and then it kind of plateaus. And it goes across cycle 30, 32, 34, 40, 40, and then it collapses. It crashes into 42. The reason it crashes into 42 is because you've hit the limit of detection, meaning this distribution carries on into the abyss, into the limit of the PCR's ability to measure it. There are samples that are being measured that are not picked up. We don't know how many because we cannot see the right-hand side of the curve, but clearly we are missing some of them. The other point as well is that the distribution is huge. Yeah, we've got almost 25 cycles, which is a lot when you take the raise that to the power of 2. Okay, well, that's one data set. Let's have a look at the Delta data set from BioGazelle. Can we have the next slide, please? This is interesting because you can see the data set is more to the left now, and this absolutely supports Mikhail Kubista's point about never directly compare CQ. Because if you move to the next slide, the data set is not unlike the Great Ormond Street data set. You get an increase, it goes across. There's possibly a peak around cycle 30, 32 here, and then at cycle 36, it drops hard. And Jo van der Sompele will tell you, he's on the call, that is their limit of detection. So again, we are missing some of the data because the PCR can only get so much of the RNA into the experiment. We don't know how much. It could be very small. It's impossible to know. Interestingly, that looks like there's a bigger peak at cycle 30, 32 that perhaps the Great Ormond Street dataset didn't have, but there's a much larger data set here. And if you move to the next slide, when you overlay them, normalizing to the mean, you can see the data sets look, I mean, I wrote to Jo last night, this looks scarily similar. And we have data now from a hospital in Seoul in Korea, and I can tell you it's not that different. And we need to review that and present that before I can show you. But the distributions of the data are very, very similar, and they are wide, very, very wide. So I would state then, arguing that we don't need to really worry about the analytical performance because the virus is at 10 to the 6 for purely analytical reasons is wrong. And so I would advise anybody who knows anybody who's making that decision to try and relax everybody that actually this is an assumption too far. And I'll talk about what might be going on there in a minute. One thing I just want to touch on, if we move to the next slide, some of you may well have heard, and this is a really, really nice article in the BBC by Rachel Sher, who's a very sharp health reporter, and gets really both sides of the argument really quite well, is that people are starting to advocate cutoffs. They realize that we may be measuring virus that's dead, or we may be measuring live virus from patients who have very small amounts and therefore are not really infectious. And so people are advocating cutoffs, which are used all the time in clinical chemistry. And people, in some of the discussions I've been involved in, people talk about cycle 25 or cycle 30. So if we move to the next slide, what that means is, if you look at the Belgian data set in the blue, is that if you had cycle 30 as a cutoff, you would remove 40% of the patients and you'd call them negative. If you use the Great Ormond Street data set, it would be 60% of the patients that would be removed. Now that may in time turn out to be completely fine clinically, but you can see just using the CQ value, you're getting a different, and the spreads are very similar. You're cutting off, you're removing different patients. So we have to understand those discrepancies. And if we're going to use CQ, we have to think in the context of calibration and trying to understand the discrepancies between the methods. However, that may still not be possible, as I'm about to show you. Next slide, please. So what this is not going to show you, because the animation's not working, is the fact that when we measure error, when we measure a biological measurement in this SARS-CoV-2 abundance, we're combining analytical and biological sources. The analytical sources are what we put in, the way we select the method, what's going on. The biological sources are not really error, probably the wrong word. They are what happens in the biology. The virus may no longer be present in large amounts in the nasopharyngeal space because it's moved into the lungs. Remember, SARS-CoV-2 is not a systemic virus like HIV or hepatitis C. It is focal. It occurs in one region and it moves, which means if you're swabbing in a place and it's not there, but it's in somewhere else, you're going to affect your method, your diagnostic performance. So if we move to the next slide, there are, as I mentioned, systems in place, and we work closely with Heinz Zeikart and colleagues who support the INSTAND study. And this was one of the first proficiency schemes that occurred in April looking at SARS-CoV-2. And what you see on the right there is calibrated qPCR, where the labs in question are calibrating their measurements and providing the values. The one thing you can see, well, two things you can see. First of all is that the methods are linear. So these are viral dilutions that are provided, and on the x-axis you're having different dilutions of viruses, and the people are asked what's present and how much is, well, they don't have to provide how much, but in this case they do. The other thing you can see is the spread of the data is huge. There's about 1,000-fold difference between the lowest and the highest. And so if you're thinking of your quantification, you have a bit of an issue there because there's a wide difference with calibration. That's with calibration. There's no harmonization. There's no WHO reference material to use here. So people are just calibrating themselves. But clearly the value assignment they're using for the qPCR is widely variable because you're getting huge differences in the amount of measured viral load. If we move to the next slide, most of the laboratories are not actually doing calibrated qPCR. And here's an example of one of the dilutions and the reported CQ values that were provided. Now, we showed you the Great Ormond Street and Belgian data sets, which differed maybe by about 4 cycles. Well, here you see much bigger differences depending on who you look at. Now, some of these are, there's some nesting involved, nested PCR, so that probably makes the cycle a bit earlier, which would explain it. But we're getting between 100 and 10 million-fold difference in the likely CQ value that's being provided. Now, the reality is probably somewhere in between. It's probably around the 50 to 100,000-fold difference if you do not calibrate the qPCR and you just rely on CQ, which means, really implementing a cutoff is going to be challenging because of the variation associated. I mean, one, as Mikhail Kubista mentioned, if it all goes through a single laboratory, if every single measurement in a nationally or internationally goes through a single laboratory, would that be possible? Perhaps you'd have a chance, but that's not reality. And so implementing cutoffs, a particular cutoff of cycle 30 is going to be problematic. What is really telling, however, is that if we move to the next slide, nothing I'm saying is new. Jacqueline Fryer and Phil Miner and the rest of the groups at NIBSC showed this in 2008, well before SARS CoV-2 was around. And if you go to the next slide, they basically showed that if you don't calibrate your PCR, you get wide variation in CQ values. So one of the things we're not doing is learning from history, and I think it's one of the things we can be certain. One of the certainties we learn from history is that we do not learn from history, and so this is slightly worrying because we know we could predict this was going to happen. Yet people are now trying to implement cutoffs on uncalibrated, unharmonized qPCR and trying to make arguments for these findings. And often in many of the papers they're finding differences because they have power. They have large data sets which show that there are, the mean values are differing maybe, but that will and cannot be applied to an individual patient because of this variation. So if we move to the next slide, while this idea of a cutoff from Professor Carl Hannigan of Oxford University is perfectly valid hypothesis, as I said, I'm not clinical, I'm not a virologist. It makes a lot of sense to at least look at that. I question whether it's actually possible because of the analytical measurement error of an uncalibrated qPCR that's not harmonized. And so the methods we're using may not allow us to do that. Next slide, please. As I wrap up. However, it's not all doom. One of the things the National Measurement Institute have been doing is looking at reference measurement procedures to try and assist and improve harmonization. And this is some results from the June-July scheme from InSTAN, which is going to be published any minute, and we're all very excited. And what you see there in the figure on the left, the red, green, and blue measurements are values by digital PCR measuring a variety of different targets. I think they're all N-gene measurements from LGC, PTB, and NIST. And those are digital PCR, including the extraction. We're getting very, very good agreement. If you saw the calibrated qPCR that's not harmonized with about 1,000-fold difference, these values are within, I think it was 50% of each other when you combine them and do uncertainty. So this, with a well-characterized extraction procedure and from that method, has the potential to support harmonization. Next slide, please. But the real, one of the real things we need globally is the international standards. And I can tell you from discussions we have with NIBSC, they are frantically working on making a WHO international standard for SARS CoV-2. It's on track, like the HIV one you see here. These are standards you can buy to essentially put values on your reference materials, secondary reference materials, that you can then use to calibrate your measurements, which basically take the chaos that I've showed you and bring everything into harmonization. And I'm informed by Mark Page from NIBSC that hopefully this will be available in December 2020. But this work, it is hard. Yes, it's tough, and to do it this fast is really challenging because calibration and standardization, it is not an easy thing to do. And so reason I make that point is when people are arguing that there may be a case for cutoffs or whatever, they really need to think in the context of reproducibility and this measurement science that I'm talking about. Because having a publication that shows when you did it with that particular patient group, it looked like it was possible is fine. But when you have people, on eminent experts in various areas of medicine or clinical chemistry, but not in molecular biology and not in qPCR, not understanding that we work in log space, huge log space in this example, and the variation is very, very large, this is a problem because people think, oh, that's a possibility. People put resources onto that, and it's never going to be possible, I don't think. Next slide, please. Well, certainly not in the current format. What I was going to show you here is the pipeline, the extraction pipeline, and the fact that a lot of what we talk about, and Mikhail already touched on this, is the extraction and the PCR bit, and many of the proficiency sets, that's all they look at. The actual sampling and the transport are the major sources of variation, which we will need to get on top of if we're going to improve harmonization. And I just wanted to show these two slides from the UK and the US, pointing out that how bad and how, we might be missing 25% of positives, or in the Wall Street journals, we may be missing a third of patients maybe being missed. In reality, at the beginning of this thing, when we didn't really know what was going on, the extractions were working, maybe people were desperately trying to get things up and running. That actually wouldn't be surprising. A 75% sensitivity from the PCR you've developed last month to work on a pandemic virus that is ravishing the world would be pretty good. So I think we need to change the narrative. The fact that, if you're missing 25% of the positives, it's flawed. It's not flawed. It can be developed, it can be improved, but it's a start. So next slide to wrap up. The Rtq-Pcr method basically has allowed the world to track SARS-CoV-2 and respond to COVID-19 pandemic. I don't think that's too strong a statement. If we took away, if Kary Mullis hadn't developed PCR, well, we can have a debate about where we might be without that methodology. There are many efforts to improve the availability and performance of SARS-CoV-2 molecular diagnostic tests, both PCR and other based methods like isothermal strategies. And there are an army of control materials that will assist in this process. But the discussions around the methodological performance and what we could potentially do with that, such as quantification or cutoffs, really need to consider what is possible from a given control material. We cannot expect, if it's not been value assigned well enough, then it shouldn't be being used for limited detection measures. Often the vendors never meant this. It's a quality control material. We need to think about the units we use, nanograms versus copies. The considerations to convert one to the other rely on a lot of assumptions, which, if you get wrong, might mean you're measuring different things. The fact that the Rtq-Pcr is a very precise method, but as we've shown, it has huge potential for bias. 100,000-fold difference when you're just doing CQ values. That is, your lab may get nice precise measurement there, but another laboratory is going to get a different level of measurement. And there are many additional considerations other than the Rtq-Pcr. You see this with the sequencing technology, you see this with the isothermal. Everybody's interested in that molecular bit at the end. Ultimately, it's the pre-analytical steps, the sampling, the transport, the stability that makes the test work or not. And it is always the most neglected bit in the development of a diagnostic procedure or preclinical research to develop a diagnostic procedure. It's always the funky technology we're interested in. But we need to spend more time on the pre-analytics. And Mikhail mentioned ISO. There are a number of studies, SPIDIA 4P, looking specifically at pre-analytics. The only trouble with many of these methods have been focused on precision medicine, cancer diagnostics. Not saying they shouldn't be focused on those, but when we looked at trying to get support for infectious diseases, it's not very popular. Well, it wasn't. I suspect it will be now. To finalize, then, investigating sources of error and improving standardization, I think, is going to be necessary if we're going to be able to assist stratifying patients based on viral burden to prime better identify patients who are infectious. However, I question whether this will ever be possible for the reasons that I've touched on, analytical reasons, that is, not necessarily the virus. Okay, thank you. Could I just go to my acknowledgement slide? So the work we do is, as I say, it's it's massively collaborative, and there's no way I can thank everybody I need to. The group at the NML are fantastic and have worked amazingly over the last 6 months to allow us to do the work we do. University of Surrey, Dan Evans is a PhD student who started with me in October, started working on E. coli measurements and sepsis, and then had to stop working in February. And he has primarily worked with the Great Ormond Street dataset and BioGazelle dataset, and it's incredible, given he's just started his PhD, looking at some of the histograms and looking at those datasets. So I'm grateful to him for doing that, and also grateful to Great Ormond Street and BioGazelle, Yo, and Catherine et al. For their support. And that work is looking really good with Chris Youngbei and Kiho Yong from the Seoul Medical Center. So watch this space. We're going to have some, hopefully some really interesting outputs in the next few weeks. Also like to thank the National Measurement Community, NIST and PTB, particularly working close with them, and Chris as well in that category, and the CCQM and BIPM. Heinz Zeikart, Martin Kammel and Hans-Peter Grüner from Instand, GBD, worked very closely with them for the proficiency testing work. And then IBSC, Claire Morris, Mark Page and Neil Arman for their support, both in guidance and also for the CCQM study I touched upon. We are using one of the NIBSC materials in that. And also I'd like to thank the people who funded the Department for Bayes in the UK, and MPIR, the European Union, funding of our work. May that continue depending on wherever the UK ends up, but we will see. I'd just like to finalize this slide the next slide, really to touch on a bit of measurement history. Essentially, and unfortunately you can't see this, I think you might be able to actually. This is Professor Desautoy of Oxford University, who's a mathematician, and he presents this really good BBC program. And essentially, he talks about the measurement and history of measurement and the meter, which kind of started the measurement. The meter started the modern metric system. And basically, in the 1960s, up to the 1960s, we could measure the distance with an accuracy of 1/1,000,000th of a meter, and that sounds pretty good. But the measurement science was necessary because, if we go to the next slide, I think it might work. Yes. If we hadn't have improved that in the 50s, then we would have missed the moon. And so often it may not be apparent what we can achieve with improving the measurements until we're able to do them. And so it's just a final point there. So final slide to thank you all for your time and thank the scientists for organizing this. And I hope the problems with the computer didn't cause too much disruption. Thank you very much.
Thank you, Dr. Huggitt, for that presentation, and thank you to Drs. Bustin and Kubista for their presentations as well. Unsurprisingly, the audience has submitted several questions, so at this point we will begin our question and answer session. Joining Drs. Bustin, Huggitt, and Kubista, as mentioned, will be Dr. Jo van de Sompele and Dr. Michael Fafel. Dr. Jo van de Sompele obtained a Master of Science in Bioscience Engineering in 1997 and a PhD in Medical Genetics in 2002. Since 2007, he's been the professor in Functional Cancer Genomics and Applied Bioinformatics at Ghent University. He is the author of more than 250 scientific articles in international journals, including some pioneering publications in the domain of RNA quantification and non-coding RNA. His H-index is 66, with over 33,000 citations as of January 2020. Dr. van der Sompele is a frequently invited speaker at international conferences and symposia, and was one of the co-founders of CRIG, the Cancer Research Institute Ghent, and chairman of the first steering committee from 2016 to 2019. Dr. Michael Fafel's training began in 1986 when he started agriculture, animal science, and biotechnology studies at the Technical University of Munich, TUM. He obtained his PhD in molecular physiology at TUM in 1997 and completed his habilitation at the Center of Life and Food Sciences, Weihenstephan, in June 2003. He became professor of molecular physiology at the TUM School of Life Sciences at Freising Weihenstephan in in 2010 and is currently a principal investigator. Dr. Pfaffel is one of the leading scientists in the field of molecular physiology with a focus on gene quantification, Rtq-Pcr technology, RNA sequencing, extracellular vesicle biology, and complex data analysis by integrative biostatistical methods and multivariate algorithms. He's the author of more than 200 peer-reviewed publications, 50 book chapters, and has held more than 250 lectures worldwide. Now, for our to begin our question and answer session, we have several topics that we could begin with, but I would like to start with something that is a little bit, perhaps a little bit directly pertaining to the presentation that we just heard. And this question, we can perhaps allow Dr. Huggitt to start, but Dr. van der Sompele will probably have an opinion as well. And that is, we talked about the curves from the BioGazelle and the Great Ormond data and the abrupt cutoff after a certain CT threshold, and that was the limit of detection. Do we expect a significant number or proportion of additional positives beyond that? In that, is the limit of detection hiding a significant chunk of the population, or have we reached the point where viral titer is not sufficient at those CT values to have a great impact?
Okay, I'll have a go, and then I'll hand over to Yo, who's done a lot more of the actual testing. I think the answer to that question is we don't really know. I suspect from the distribution that there is RNA present in samples that are negative because you didn't get it into the PCR because there wasn't high enough titer. Now, again, handing up my non-clinical virology status here, that may be dead virus, such a low signal that it is inconsequential. However, it could also be a poor extraction procedure that means that we've got a lot of virus that was actually there, and therefore it's a lower signal. What we can tell you, and I think Yo can challenge this, but certainly from the Great Ormond Street data, it is that their methods, and I'm pretty sure the BioGazelle, they're close to single molecule detection because they're so well developed. They do this all the time, so we're fairly confident that they are. They're measuring tens or less than tens of RNA molecules. But what's on the right hand, what's greater than the 36 or the 40, depending on which you look at, is unknown. It's unknown. Is it relevant? Don't know. People, I think in the clinical space would argue no, it's not relevant. But again, we're now in that point of the fact that the SARS-CoV-2 test is a yes/no test, and at the moment, while people may be starting to apply CQ thresholds or thinking in the terms of that, I think I've argued quite strongly that they shouldn't. And again, actually, just to clarify, if someone comes in with a quantification cycle, CQ a result of 15 versus 35, then I guess generally people are going to think, well, there's an awful lot in the case of the 15, and perhaps the 35 is low. And people are doing that, and they are seeing that, or it's like HOLI. But we've also showed you that you can get huge variation. But I guess it's in the same lab, then you can know that. But to actually have take a lot of patients and have a cutoff and put them all in there, that is worrying. In short, we don't know. There is going to be false negative. There's going to be RNA present that the PCR did not take. There's going to be patients who have SARS-CoV-2 infection, undoubtedly, in that data set that didn't get into the PCR reaction, whether it's because they had very low titer or because the extraction didn't work, or a combination of both. What they mean clinically is not for me to say. What do you think?
I agree, Jim, with your statements, and I think also Mikhail Kubista referred to that. I think the qPCR is the easy part. If the RNA molecule is in there, if you have a good assay and a good master mix, you will be able to detect it. Of course, not be below the theoretical limit of detection of 3 copies. Above that, in principle, you can detect it. I think the challenge is more upstream, where the RNA extraction is not only variable, but you lose sensitivity, and then even more upstream, the sampling part is not very well controlled. So by focusing too much on the qPCR aspect itself, I think we don't do justice to describing the sensitivity and specificity of the entire workflow method.
Yeah. Can I say something? Can you hear me? Hello?
A little bit faintly, but
I completely agree, and that is why we have decided to focus on the sampling, the ease of sampling, particularly if you're trying to self-sample through a nasopharyngeal swab, you're not going to be very successful, I think. And so other methods and other samples are going to be very important. And also the concentration of the virus whilst you're extracting, I think, is important, because as everyone has said now, the qPCR bit is probably the easy bit. It's getting to the qPCR that is the problem.
all right. Thank you for those responses. For our next question, perhaps Dr. Bustin can address or have the first word. We talked during the course of the presentations, we brought up the topic of doing multi-target analysis and multi-target detection and subsequent analysis based on that. And in Dr. Huggitt's presentation too, he talked about how different testing protocols, different organizations around the world have chosen different targets for their testing. So how does one decide what target to look for? How does one decide how many targets to look for? And is there a minimum number or a maximum number?
That's obviously a very complicated question. I think one is too low and 5 is too high. So we go for 2 to 3. I think that's probably, I think 2 is reasonable. One of the things that we did when we designed our tests was we tried and looked at RNA structure to see which parts of the RNA might be more or less stable. We also looked at the distribution of mutations as they became available, to see if there were any areas of the genome that were more or less stable. But I think it is a question of suck it and see, develop a series of assays, and then keep on monitoring the database to see whether mutations are appearing, and hope that, like in our case, we have mutations in the forward primer that doesn't seem to matter, whereas others have had mutations in the reverse primer that affects the RT step.
May I comment as well on the amount of targets?
Of course.
I agree with Steven's opinion, and I would think 3 is the optimal number. 1, to have a sensitive assay. 2, to confirm that sampling is done by humans, and 3, also to confirm the positive results. So 2 SARS genes and 1 general human gene. I think that's the optimal combination for an assay setup. And it's not too complex, it's not too multiplex, it's easily achievable. Thank you. Could you hear me?
Yes.
Okay.
I was just wondering if anybody else wanted to add to that.
It's Michael here. It's complicated for many reasons. One is the cost. It's more expensive. In the beginning, we did measures up to 3 genes for validation, but not today. Not in mass testing. But it could also be a matter of sensitivity, because if you test 3 targets, you are essentially increasing the sensitivity below LOD.
I have one comment, if I may, following on Michael Faffel's point about internal controls. There's a different choice. People have and do use spikes, synthetic or Non-SARS-CoV-2 viruses are often used. Certainly great industry to that. Some labs are also advocating human RNAs, like RNase P, which look like they work, when I've seen the data, look quite well. However, we are now looking at potentially highly abundant RNA molecules, and so you may find that you get a positive result from your control, depending on what concentration it is, but you've actually only extracted 1% of the RNA, and the lower signal virus is gone. So we have to be a little bit careful about the concentration at which the control is that we're measuring.
If I may follow up on this, and I believe there was also a question from one of the attendants, is the relative merits of an endogenous RNA control versus spike-in control. And actually, there are two schools of thought, and actually also in practice, two different strategies. And I was intrigued by Steven Bustin's proposal to actually do both, because they both are valid and they measure something different. I particularly like the spike-in control RNA on itself because it's very well controlled. There is built-in truth. You add the same number of molecules to each sample, and if your extraction works well, there are no inhibitors, etc., then you should come to the same quantitative result for your spike-in control. So in terms of workflow control in the analytical lab, it's very helpful. Of course, you can still have the situation where the sampling from the patient did not occur well, and the spike-in control will not tell you that. So human RNA is useful to assess sampling at the patient level. However, I'm a little bit appalled by the low bar, amongst others by the CDC and other institutes as well, that simply state you should have a positive signal. I think that is a really low bar, that you only should have a positive human RNA and then you can trust the result.
So I think we've touched on this subject a little bit, but I was wondering if anybody else had any comments on what an ideal positive and negative control would look like and how they would be implemented into the testing.
If I may start, I think we have to differentiate internal controls from sample or workflow run controls. We recommend positive and negative controls to be included very early in the workflow. So also undergoing primary sample transfer, RNA extraction, Rtq-Pcr, to make sure that the entire procedure, you control the batches, the lots and the reagents. And in addition, of course, an internal control, an internal positive control, where we have the choice, like we discussed before, spike-in controls versus endogenous RNA. We tend to favor the spike-in control, but I do appreciate there are different opinions on this one.
Actually, to add to that then, Yo, on the controlling of the experiment, there's also the negative controls, which you touched on. And I just wanted to use this opportunity to plug, nobody in the speakers are going to object to this, because all 5 of us are on this article in Clinical Chemistry we've just published. It's just come out, talking about contamination and false positives. I didn't really have time to talk about that today, but Mikhail mentioned, and Stephen may have done, that a negative result should be that. There should be no result. There should be no signal. And there are numbers of laboratories who have published in peer-reviewed literature stating that there is a background signal. You might have an ELISA. This is contamination, and it's contamination probably from the oligo houses, and the fact that everybody who did PCR 6 months ago who didn't have a category 3 laboratory to handle the virus made the molecule. And so all oligo houses are likely to potentially have this molecule there, and that may contaminate the reagent. So it's important to have a large number of negative controls, certainly when you're starting up and having new reagents, and to have negative controls throughout your experimental workflow, and to be cognizant of the fact that if you're getting large numbers of very high signal, very high CQ signal, then that to be fair, that may be due to contamination. And there are other sources of contamination, like the fact you might have a very high viral signal that then is processed near some negative samples, and they will then just cross-contaminate, because if you have 10 to the 8 copies of virus, which in some of Yo's samples I think you probably have, then it's very difficult not to contaminate and cross-contaminate. So to monitor the position of the actual the results, if you have a cluster of results in the same place, well, that may be because they all come from the same hospital, but it could also be because of the positioning on the plate. So just beware of the negatives as well.
I agree with everything that's been said. I just would like to expand perhaps a little bit on what Yo just talked about, which are the assay controls. I think one of the most important controls you can have is a artificial spike, because it tells you something about your assay. And I think an internal mRNA control is useful, but not necessary. I think it is useful to know that you have got some RNA there, but you may not always get mRNA. So on the other hand, the artificial control tells you that there has been a problem with your test, inhibition, for example, or you've lost it during the extraction. And I think that is the crucial control that needs to be included, a positive control that needs to be included in every test. And that's why we've got it in there. And we have a CQ of 25, which I can't quite remember what it is now, but that comes up consistently. A local hospital, the samples we've extracted or they've extracted come within 3 CQs of 25 cycles every time. So we know that their extraction method is very good.
If I may add to that, Steven, talking about controls, I think it's very important to use positive controls, whether they're endogenous controls, like spec-in controls, or sample controls, workflow controls, is that they have as low concentration as possible, ideally around the LOQ, because it's there where you do see differences in reagents, in performance of your instrument. It's all too easy to detect 1 million copies. You will not see deviations of your workflow. However, at 100 or 1,000 copies, that's where you start noticing that there may be something wrong with reagents or your workflow.
Yes, thank you. Also, just as an aside, our internal control, we've engineered so that it has the same primer sequences as C-June. So we have one set of primers less, talking about problems with multiplexing. So 2 different probes picking up 2 different targets with the same primer.
One comment just to come in, I noticed in the question panel is that someone stated that the CDC recommends CT greater than 40 in the NTCs as being acceptable. If they do indeed do that, I would disagree with the CDC. I think this is a mistake, because you are starting to allow signal. Now, depending where that signal is coming from, if it's you've got to look at that. But if it's PCRs being amplified, you're getting an amplification curve, which you can see if you go to this clinical chemistry paper, I'll plug again, we can see examples of that. Then it shouldn't be there. And so you have to question is why. Now, there may be a situation where you have contaminated the lab so much that that's all you can do. That's a different discussion. But that will limit your limited detection and what you can measure. But I realize there's a discussion between people may say, well, don't let the good be the enemy, best be the enemy of the good. But we should be able to run PCRs with NTCs that are negative. That's what we do on a daily basis.
Jim, I couldn't agree more. And
you've done 100,000 plus tests.
Yeah, so I couldn't agree more. And if not for the reason that mentioning a CQ as a threshold is, for all reasons discussed by all of you today, doesn't make sense.
I think we're going to have to wait for the biology to catch up with us and tell us how relevant very, very low levels of virus are in.
Yeah, I completely agree, accepting that the methods being used to measure the biology are inherently variable and therefore will influence how well you can actually make those measurements in the first place. I mean, that's my point, I think. Unquestionably, there will be clinical reasons and whatever where actually we can have they may be able to have a cutoff. But at the moment, those decisions and a lot of those studies are being made using methods that are not reproducible. I guess that's my point.
And a related question from one of the attendants was a reference to an article that was then featured in New York Times, I believe. I don't know the exact details, but it's in the Q&A. And that mentioned that 30% of the results or 70% of the results may be false positives. And I really find it a pity that it's framed as false positives, because if you read the article well and the study very well, it's not about analytical false positives. It's about the question, what does it mean to have a low positive result. And indeed, the question is, are these patients infectious or not? And that's a very relevant question. But that's beyond the analytical discussion, whether it's a true positive result, low positive, but it may have no consequences.
Is there anything else anyone would like to add? If not, we can move on to the next question. And one of the overarching themes across the presentations today was the importance of not just looking at in-lab analysis, but also pre-analytical steps. Now, there are many challenges to standardization and harmonization of pre-analytical steps, ranging from collection. There are differences in terms of logistics, geography, population density, so on and so forth, to the point where it may not be possible to fully standardize, fully harmonize everything, especially when we're talking about things like travel time and transit time. Is there anything that can be done? Well, first of all, tell me if I'm wrong about that. And second, if I'm right that true harmonization is not practically achievable, is there anything that can be done within the lab to perhaps mitigate or limit the effects of pre-analytical differences? And I will ask if Dr. Kubista could kick us off.
It's difficult. I mean, using endogenous controls is a way to test for the pre-analytical variation. Steve Bustin was mentioning that, like a human RNA control, that gives you an idea about issues that may have appeared. I think that's the best thing to do, really. But I have also a comment to Jim and Joe's data, where you are showing the statistics of the CQ value. You guys are interpreting them as a biological variation. I'm sure there is a biological variation, but have you considered that there could be actually a variation due to the person doing the sampling?
So, Mikhail, yeah. So, unfortunately, my animation wasn't working when I showed that yellow bar to point out very point that we are showing everything. Yeah, and so if that came across, then that was a mistake of mine. That the data from Yo's group and Great Ormond Street it's everything. Yeah, and we do not know. One of the problems is a lot of the analysis and a lot of the conclusions are made is not realizing, you're very right, the fact you have analytical sources of variation, sampling, extraction procedure, whatever, as well as what's happening biologically. And what the measurement research and the measurement scientists are doing is splitting them. And so trying to look at describing again, though, we're trying to look at describing the performance of the extraction methods, how you can harmonize that process, try and reduce that. The work I showed you from Instand is just tubes of cultured virus. So there is no pre-analytical sources of error. And even then, we're getting 50 to 100,000 variation in CQ. So it is large. I suspect not an insignificant amount of variation is actually from the technical, the sampling, the processing. That. Yo, what do you think?
Yeah, true. It's a compound variability of all these kind of sources, not to mention the timing when the patient was sampled. Maybe the biggest is there at which time point do you capture the patient? We've seen that in Belgium with the second search, is that we saw far more higher concentrations of viral loads, CQ10 and below, and because we probably have detected these contact tracing individuals very early, and that's why they had massive viral loads and start being some kind of super spreaders. And in May, that was a very different picture in Belgium, where we had many very low positives. So same region, but just a different phase in the pandemic.
May I comment about the pre-analytical stuff?
Of course.
We tested this in the beginning, around March, April, because we are also worrying about the pre-analytical loss of viruses. There are papers out which are documenting very well if the living virus is on, let's say, copper, it's gone for 4 to 6 hours or 8 hours, something like that. On a piece of paper, on cartonnage or something like that, it's gone after 1 day. And if we are thinking about these swabs are mainly out of kind of cotton stuff, paperwork or something like that, how a virus can be stable there for more than 2 days if you're using a dry swab. And then, of course, we have the liquid swabs with unknown buffers, and of course, we have the RNA protecting swabs. This is might be the best, where they really the virus is in a protecting and an RNA stabilizing solution. Then it will survive the travel and maybe more than 1 day to the lab. But to be honest, if you sample on a Thursday or Friday, send it to the lab, and it sits there over the weekend, or even it's analyzed over the weekend, and it's traveling for 2 or 3 days, even if it's hot outside, I'm pretty sure there is no virus in. So hence we spiked with armored RNA. We did spiking experiments for our dry swabs with and without sputum, and we could not get any conclusion out of these data, because on one swab we had almost all remaining, it was stable. On the next swab, everything was gone. So it's a huge variation on the virus stability and in terms of time and from person to person, even then. And even if you standardized the sputum, saliva, it's not reproducible at all. And even the amount on the swab, we measured how much amount of saliva is on each swab. It differs between 40, 60, 80, up to 120 microliters, because also the swabs are different in size. So it's very hard to standardize this and evenly to get an analytical answer out of it.
My solution is totally impractical and much more long term. We are trying to, in my opinion, the solution is going to be an inexpensive device that you spit into and that extracts the viral RNA from the sample and then immediately reverse transcribes it into DNA. And you do that in situ, and then you've got DNA. And you have a cheap device that can be done locally.
I fully agree, because as soon as the virus is dried out, it's gone, it's dead. And in the sputum sampling, like Steven is promoting it, you still have 0.5 ml, 1 ml, or even more, and it's always liquid. And there the virus can survive over, let's say, multiple days. But on the other hand, you've shown very clearly that you have a inhibiting action on RT and PCR. So yeah, but sputum sampling, I think it has a great future.
It's Michael. I have a comment that related to pre-analytics. I think it's very, very hard to standardize pre-analytics for quantitative analysis.
Yeah, that's what I said. Yeah.
Yes, that's correct. But be aware that most of the testing today is actually qualitative. So essentially, all the kits that are approved are actually qualitative. It's only negative and positive. Sure, you're getting CQ values, but one has to be very, very careful about the quantification.
I agree, Michael. I agree, but we're talking about sensitivity. Steven and Jim showed some data. we even in-house have different assays with 5 molecule sensitivities, 1,000 molecules, or in between. But if you count these different sensitivities of these assays and count on top the loss through transportation or pre-analytics, how can you trust the data anyways? That's a big problem, I think.
Yeah.
This is showing nicely, actually, the role. Sorry, Michael.
Sorry. At least we should bring the virus as much as in a living status to the lab. Yeah.
So this is showing quite nicely. SARS-CoV-2 is showing a spotlight how important some of these quantitative metrics are to qualitative measurements. The extreme being the limit of detection, but with molecular, the molecular measurements we do on we're trying to, all these guys and a lot of people here trying to do, looking at gene expression or looking at, cancer biomarkers or whatever. These factors apply too. Some things are easier, perhaps, like, viruses that are systemic. I mentioned the HIV, where it's established. But many of these challenges are actually, they're not just SARS-CoV-2. It's just we're happening to talk about them now. And I think it's, we perhaps use, not as an opportunity, but to learn from this because it's under such a tight spotlight, and perhaps, improve the wider molecular diagnostics, because these are the ranges. This is what we deal with. These are the distributions, and people trying to use qPCR in this way.
When it comes to qualitative testing, I think there are two things that are important. One is to really avoid false positives, and Jim already discussed that, where the major problem is the contamination. The other is the pre-analytics to have a certain minimum sensitivity. And I actually believe that now when we have the armored material, that we should validate entire workflows and essentially sort out those steps that more or less compromise sensitivity. Like you, Michael, were saying, for example, limit the storage time at room temperature to whatever, 12 hours, to have these kind of indicators. This, I think, is step one.
Yeah.
Thank you for those.
What is the conclusion out of these pre-analytical steps? we need better sampling technologies, sampling and virus or RNA protection technologies. I think this is the most important thing.
Right. Thank you for your answers and your comments to that question. Our next question is, now that we've almost 9-10 months now, if we start from the beginning in Wuhan, now that we're 9-10 months into the pandemic, does the prospect of the potential secondary infection or the presence of antibodies, does that impact SARS-CoV-2 testing? Does that confound the protocols and standards that have been kind of established based on a population that didn't have these potential confounding factors? I can ask Dr. Bustin to get us started.
No, I don't think so. We are testing RNA, and we are looking for a molecule that is present a couple of days before symptoms start and a couple of weeks until after the symptoms have disappeared. So what happened 3 months ago shouldn't really impact what happens next month. I think our testing procedures have become better. I think the testing infrastructure is the thing that's the problem. As we discussed, it is the sampling, it is the transport. In the UK, some people are supposed to travel 200 miles to get their tests. So all of these things are going to be far more problematic than the test itself, in my opinion. I think we are learning more about antibody tests. We're learning more about antigen tests. I think they have their place. But as we're learning, the virus, the immune response is not really behaving the way we might have predicted it. And so if someone is antibody negative, that doesn't mean they have not had the virus or they have had the virus. It just means that in this particular individual, the response has been a short term, and he still may be protective. So I think there's so many things we just don't know about this that I would be reluctant to comment too much on that part. What I would say is that the PCR tests themselves should not be affected by any of this.
All right, thank you very much for that answer. And I think I would like to kind of close with one, hopefully a good discussion, one final good discussion. And Dr. Bustin, you kicked us off with the notion that testing has been in shambles. And you mentioned just now in your answer that we've come a long way in terms of developing a robust testing infrastructure and basically establishing standardization and basically figuring out what we're supposed to do a little bit. Where are we in terms of developing a proper SARS-CoV-2 testing infrastructure? Does it differ in different parts of the world? I mean, it does, but how much does it differ in different parts of the world? And what would the foreseeable future look like? It's a hard one, I know.
Are you talking to me? Well, I think I'm somewhat cynical about this. As I said in my talk, I think ultimately politics trumps science. And what we're seeing is not just in the UK and in the United States, we're seeing the whole pandemic being used for political purposes. And so ultimately what will decide how things are done and whether things will be done properly depends on who is in charge of the political decisions. And so far we have seen so much politicization and incompetence that I'm not terribly hopeful. Having said all of that, I think if we continue to plug away at trying to produce tests and procedures and workflows and interpretations and biology and so on that is based on fact, then we might be able to come out of this. But yeah, I'm somewhat dismayed and despondent at how we are behaving as a global community.
Can I add to that, Stephen? So I half agree with you, but there's another problem as well, and it's the problem that the five of us and many others in the Mike community have been trying to drive for the last 20 years. And it pains me to sound like I'm actually defending the government in the UK, who I certainly didn't vote for. The scientists are at fault. Yeah, there are expectations, and I listen to some of these experts who are experts in A, B, and C on the news, and they talk with great knowledge on A, B, and C. And then when the reporters ask them about D and E, they carry on with great knowledge that they clearly don't have, certainly not in the molecular space. And so we have conversations with people talking about limited detection of the PCR and expecting things like the limited blank and what is the background signal, and it's nothing. What do you mean it's nothing? And comparing, like we have a new assay coming in and they compare it with the old assay. It's like, well, no, it's a PCR. You know, PCR should be measuring near single molecule. You know, that's how you do it. And people are not basically applying rudimentary molecular biology knowledge that you guys do on a daily basis. And these experts are not coming to molecular biologists to answer these questions. And, you know, great example in the US where a certain institution that was key for making the tests made the product of the PCR next to where they were running the PCR. I mean, we know that's going to cause certain problems with a method that is able to measure one molecule. So there's some really basic errors being made amongst the scientific community, which is very frustrating. And you know, and many of these people are advising the government in question. And so, you know, at the risk of not entirely disagreeing with you, Stephen, I think, and while the political argument is, I think, valid, some of the decisions that are decision makers have made has been on science that is not really, the advisors have not got it right. And basic stuff now. I'm not talking, talking metrology or the whatever. I'm talking the basic stuff.
I completely agree with you. And we just have to look, as you say, we've done 20 years, we've been trying to get people to use qPCR properly, and they're still not using it properly. So yes, I think your point is very valid.
I do hope that we will have learned something from this pandemic, which has not ended yet, unfortunately. And that, of course, I think most of us agree that qPCR has shown its tremendous value in trying to, as one of the elements to keep the pandemic somewhat under control. I do hope that governments will learn to build an infrastructure to have it available for quickly deployment. I think that's key in managing this, but also future pandemics. I think contact tracing is an element that needs improvement in many different countries, and that will allow us to do very quick testing and quarantining the right individuals, working with drive-in testing, local places that people don't need to go far away, which is problematic in itself. And especially for the sampling part, it's really appalling to see that we've been using sampling devices from the previous millennium that are intended to keep the virus alive. Well, we don't want that for all kinds of reasons. We want to protect the RNA, but not keep the virus alive for safety reasons. So I think technological developments for much better sampling, be it nasal or oral or saliva, I think we can discuss about that, but more standardized and safe collection devices is important. And perhaps also considering pooling as an alternative for surveillance. There are some limitations, but also some advantages of pooling. And I think these are all elements that need to be made part of a grand scheme and a strategy that any government needs to have ready for future response to similar pandemics.
Yep, I agree.
And so I suppose to close, as we move towards what people are forecasting as an upcoming second wave and the winter hits, are we in a better place than we were 6-9 months ago?
I think that may depend very much on the country. I think in Belgium we will be, or at least the plans are there. If you see in terms of testing capacity and united forces, it's impressive what the government has done, despite all problems and criticism that some of it is justified. I think just to see that the testing capacity is so high that in principle any individual with a potential seasonal flu can and will be tested. I think we're probably very well prepared. And it's a huge logistic undertaking, but it's about preparation. And I think the winter season is not there yet. We still have 3 to 4 months to go. So if countries have not prepared themselves, I think it's not too late to set up all the logistics and the testing to be prepared.
It's Michael here. I think that we will see an improvement in the workflows, particularly faster workflows, including point-of-care devices. And that can be quite important to test somebody in an hour or so. For example, at workplaces or care houses, emergency care, and so forth. This I expect will be very important in the near future.
Just to add with that, I think the answer is yes. I mean, we are, you think where we were 6 to 9 months ago, whether it's us, the diagnostic community, or the medics fronting this, or just general, we had no idea what was coming. Well, we now know. And I think it's an opportunity to stand at this, and hopefully capitalize on the fact that we are all working hard to, and these challenges, difficult times require challenges, and to meet these challenges, and hopefully we will the whole testing, the molecular testing, will actually benefit beyond just SARS-CoV-2 testing. I know you're now doing infectious diseases, which is always a good thing.
Are there any other comments that anyone would like to make? If not, that is all the time we have for today. I want to thank everyone for their attendance, and if they have any further questions, please consider reaching out to the speakers directly. And as a reminder, the webinar will be archived on the scientist's website, and you will receive an email notifying you when the on-demand version is available. I would like to profoundly thank everyone who took the time to join us today, and particularly those of you in the audience who shared your questions and your comments. On behalf of the scientist, I would like to thank our panel of speakers, Dr. Steven Bustin, Dr. Jim Huggitt, Dr. Mikhail Kubista, Dr. Jo van de Sompele, and Dr. Michael Fafel, as well as our sponsors, Millipore Sigma, PHCBI, Acro Biosystems, Biosearch Technologies by LGC, and Codex DNA. Thank you, everyone, and have a pleasant day.